Thank you very much, Dr. Shaji, for nice introduction, and thank you for inviting me to talk and share my experiences in this group. Welcome and good evening, good morning, and good afternoon to all the participants. I can see there are some very senior homeopathic physicians. Dr. Ismail Seth is here, Dr. A.K. Gupta, and many other doctors are there. So my respects to all the senior people and my respect to all the homeopaths who are taking time and trouble to learn something about some possible solutions whereby we could help the humanity during this pandemic time. So I have been suggested to talk about COVID-19 nosode, and I have not prepared any PPT, but I'll talk uh, extempore my experiences because I have spent a lot of time and lot of energy into the research with this COVID-19 nosode, and of
Okay.
course with many other nosodes in last 20-25 years. So, and I'm currently we are also working on creating more awareness about this nosode and the potential that probably it bears in helping the humanity. So I would like this meeting to be a little more interactive towards the second part, whereby I would invite questions from the participants, and I'll talk based on the interest level of the participants. Before that, I'll briefly give you an idea about what this COVID-19 nosode is and how we developed it and what it has done so far and what it can possibly do in the coming time. So, friends, going back somewhere February 2020, so February, yes, 2020, year and a half ago, I was at a conference in Kochi. And at that time, the pandemic had just begun in India. We had reported some 2 cases of COVID, first time ever in Kerala, if you recall
Wow.
that. And that
Yeah.
was the time the conference was there, and I was speaking at the conference. And I mentioned in my talk at the conference that we must immediately start working on making the COVID nosode, because COVID nosode could possibly help this pandemic. Of course, a lot of my colleagues and my friends, they just laughed it away. They said, "Oh, what are you talking, Rajesh? I don't think this this pandemic will last long." Some people said. A very senior scientist from the government also said that, Rajesh, I think this this pandemic will go away in 2 or 3 months' time, and maybe some vaccine will come very soon. And I don't think there is a need for any nosode. These were some observations made by some of my senior colleagues. Anyway, as a habit, I don't listen to everybody every time. I do listen, but I may not follow what everybody says. So I immediately coming to Mumbai, we started working on the nosode, and I'm very happy to share with you that precisely 1 year ago, that was 22nd of May, we developed the first variant of COVID nosode at Hafkin Institute, Mumbai. Hafkin Institute is a very famous virology institution in India, 125 years old, which also makes the various vaccines, including BCG vaccine, MMR vaccine, and so on. So, and in the past, we have developed a couple of nosodes at Hafkin Institute. So our scientist friends, they are familiar with nosode and its development process. So, and I'm happy to say that this was, in my view, the first ever developed or scientifically developed COVID nosode in the world that was made on 22nd of May 2020, year ago. Now, this came, in fact, sometime earlier than the COVID vaccine which came in the world, and that is no new for homeopathy. Yeah, if I take you to the past, uh, uh, Robert Koch found Mycobacterium tuberculosis organisms somewhere in the year 1882. But very interestingly, the homeopathic nosode tuberculinum was developed about 7 years before the Mycobacterium tuberculosis organism was discovered. So nosode in homeopathy was ahead of even the recognizing the organisms itself in the microbiology. Similarly, the first homeopathy nosode from rabies virus was developed in 1835 by none other than Constantine Herring, and that was precisely around 35 years or maybe 40 years before the anti-rabies vaccine uh, was developed. So homeopathy has remained ahead in in the development of vaccine-like preparations or nosodes. So similarly, this time too, COVID nosode probably stepped up a little ahead of the the COVID vaccine. However, having said that, I must also say that homeopathy has failed in conducting scientific research in the area of nosodes. So as a result, homeopathic nosodes have not really grown in last 100 years. And homeopathic nosodes have not been identified as a possible therapy, either prophylactic or the therapeutic measure. And that is because of the lack of research. However, now is the time that if we do more scientific research in the area of nosodes, then probably I personally believe that this is the right time to advocate homeopathy and let the world know about the potentials of homeopathy as a as a modality for the treatment as well as prophylaxis. And nosode could be a gate opener in my view. And if we have successful research with the nosode, especially COVID nosode, then I'm sure we can draw the attention of hundreds and thousands of mainstream scientists who could uh, take up this work at a much higher level, and we could explore the hidden and untapped potentials of homeopathic potentized medicines. So with that, I think I must tell you very briefly. The first version of COVID nosode was developed from the clinical sample of a patient suffering with COVID. Now, it was not very simple. It is many homeopaths believe that making a nosode is a very simple thing. You just take the saliva or the sputum of a patient and put it in alcohol, give some strokes, and the nosode is ready. Well, that is exactly what happened 135 years ago. But now, when we have to make a nosode, there is a huge process because the microbiology has evolved, as we all know. There are safety measures to be taken care of. So we, first of all, we require what is called as BSL-2 facility. That is a biosafety level 2 facility in a virology laboratory. And before you develop the nosode, you require A, the ethical permission from the registered ethics body, and B, you require certain biosafety committee permission. So with these two major formalities, and of course we require an approval from the scientific advisory board. These are some of the formalities which are very important and time consuming, post which you have to have an access to a BSL-2 plus facility. So our facility at Hafkin Institute was BSL-2 plus facility, which was practicing BSL-3 modalities. So that kind of stringent parameters were followed in the making of the nosode in that laboratory. Because at the end of the day, when you develop a nosode, you are handling a very serious biological material, that is the virus. So with all these stringent regulations, the nosode was prepared. Post which we had two more variants of the nosode. We also developed a nosode from inactivated virus, and the third variety of nosode was prepared from the spike glycoprotein, the protein material in the virus. So these are the three nosodes which were developed in the month of May and subsequently in July, early July. And currently we are also working on developing the another variant of the nosode based on the current mutations and current strains which are available and which have been found to be more serious, as you all know, in certain parts of India. So we are trying to develop that, and I think hopefully in a short time we should be ready with these nosodes as well. So that is all about the development of the nosode. Now that is not alone. That is not enough. I mean, uh, we often believe in homeopathy, and we like to believe that once you have a nosode, the job is done. And the nosode, and we have a lot of presumptions. We like to presume many things in homeopathy, that if the nosode is made, it will straight away prevent the disease, or it will cure the disease. And you all know that it is not that easy to prevent and cure such a deadly viral pandemic. You need to have scientific studies at every level to prove anything or to claim anything. So we decided to follow certain universally accepted guidelines, of course. And one of the, you see, the most important thing in the development of the nosode and the use of the nosode for the humans is safety. Since it is prepared from the biological material, so its safe use in humans is the most important thing. For getting any approval from the government body, safety criteria must be met with. So our primary concern was to establish the safety of the COVID-19 nosode. So we conducted an animal toxicity study. Now this animal toxicity study, there are guidelines which are prescribed as per what is called as OECD guidelines. So these OECD guidelines are the international guidelines for animal testing for safety of any new drug. So this study was done on 2 different species of animals, and through one accredited, recognized third-party laboratory, which was having a GLP facility. And this laboratory conducted the study on animals to find out the safety level of this nosode. So that took almost more than 2.5 months. And the finding was that COVID-19 nosode was safe in animal study. Now, once it is safe in animals, it is likely to be safe in humans, because that is what is accepted. That is an accepted guidelines. This was a very important step that we had to do. After conducting this, by the way, I must also tell you that COVID nosode was probably the first nosode ever in the entire world which underwent animal toxicity study as per the OECD guidelines. So there are many things which happened for the first time during the development and research of this nosode. And I'm happy to say that this happened in India. So India should take the credit for having developed this nosode with this particular stringent guidelines. After having the animal toxicity and safety study, the next and important step was to conduct a human safety study. Because after it is proved safe in animals, it has to be proved safe for humans as well. So what is called as a phase 1 study, we conducted a phase 1 study on healthy volunteers. Again, there are ethical approval guidelines and there are other stringent guidelines which were followed, and with the CTRI registration and things like that. I will not go into the details of those technicality, but all these formalities are very important parts of the scientific studies that we homeopaths must now know, because now is the time, if you want to talk scientific language and if you want to tell the world what kind of studies the nosodes have undergone, we all must be familiar with the regulatory requirements for developing the nosodes for any new homeopathic drug. There's a reason I am coining some of this terminology which must be used by the homeopaths more frequently in the coming time. So in the phase 1 study, the study was done on 10 healthy volunteers with the prescribed guidelines to see whether this nosode was safe for the human use. And this study went on for 60 days, and we had an extension for 30 more days, approximately 30 more days. So the study went on for 90 days. And we had 2 very interesting outcomes of this study. Of course, the first outcome was that COVID-19 nosode was safe for the human use. So its safety was established once and for all. That was the primary requirement of the use of a new drug. And the second very interesting outcome that we achieved out of this human study was that 90% of the volunteers who consumed the nosode during the study, they had demonstrated some increase in certain interleukins or certain cytokines in their blood reports. And those elevated reports were suggestive of some activation of the T cell. So it was called T cell activation. And with this 90% of the volunteers having shown increased in interleukins, 70% of them, they had an increase in what is called as CD4 count. So the volunteers proved that the COVID nosode was not only safe, but it has also proved that they produced a certain increase in the CD4 count, which was suggestive of T cell activation. That was suggestive of innate immunity activation. So this was a very interesting finding because this is what we wanted. We did not expect that in the study, to be honest. But then this was the outcome of the study, where we found that the nosode was capable of inducing certain immune response. With that findings, our next step was to see its efficacy as a prophylactic measure. So during those years, that was during those months, that was somewhere in July onwards. If you remember, we had a very stringent lockdown in the city of Mumbai. Mumbai was one of the hot spots in the country, and the municipal corporation had several quarantine facilities in the city of Mumbai, which were housing those people who were exposed to COVID infected patients, especially from the slum areas. So we had a facility of housing around 1,500 to 1,800 people on a regular basis, and we conducted a study, an observational study amounting to phase 2 study among 2,233 high risk individuals. So these 2,233 people were those who had an exposure to COVID infected patients. So in this study, we conducted a multi-arm comparative study. So it was a 6-arm study in which we compared the efficacy of nosode with arsenic, bryonia, camphor, placebo, and a combination medicine. The combination medicine was arsenic, bryonia, gelsemium, and influenzimab. So these 6 medicines, 6 arms were there. We had a very good chance to make a comparative study showing, try to understand and elicit the efficacy of these homeopathic medicines against placebo. The study went on for, I think, almost 3 months or more. And our findings in brief include that the nosode had shown 62% efficacy over placebo. And that was very interesting because even as per the WHO standards, if any medicine or any vaccine, if it shows efficacy more than 50%, then that is considered significant and it requires some kind of usage and permission or approval. So nosode had shown 62% efficacy. Bryonia also had shown good efficacy, and then arsenic has shown efficacy. In this study, camphor did not show any efficacy better than placebo, and even the combination medicine did not show great efficacy, though it was better than placebo. So, of course, all the studies what I mentioned, there are research papers getting published or either published. So you can go and read the details of these studies. So now these findings were interesting because by this time we established that homeopathically prepared nosode from COVID-19 virus was not only safe, but it was also effective, that too as a preventive measure. And I would like to remind you that this happened when the vaccine was not yet made available in the world. in India at least. Vaccine was just in the making, and the nosode had shown that efficacy. So this was good. However, and then of course, lot of I would probably use the word struggle if it is not misunderstood. A struggle went on from those days, whereby a lot of my time and energy went into writing to the government agencies, various government bodies, right from the ministry, center, the health ministry, the Ayush ministry, the government of India, the Prime Minister's office, WHO office, state government, and so on, requesting them, presenting them with the documents, with the data, and requesting them to allow the use of this nosode as emergency medicine, because nothing else was available. Please remember, these were the days when the vaccine was not available. We had nothing. We had regular homeopathic medicines, of course, as genus epidemicus, as it is called, like arsenic, and other herbal medicines were used, and the vitamins and other medicines substances were utilized. So, however, you know, uh, in order that the government listens to you, you require a big lobby. Government doesn't listen to individual researchers and scientists. But that apart, that was not a great news. But anyway, I also have some good news which I'll share you in the course of time today. So having said that, of course, we continued using this medicine, but in a very limited manner, because we did not have license to use the medicine without the consent. So we used it clinically on some of the individuals whenever we got a chance, by taking the informed consent forms and such documentation. And I can say that we observed uh, very good preventive capabilities of this nosode in number of people. In fact, we had many cases where families were infected with COVID, families of 2 people to 5 people, or even 7 to 10 people infected with the problem. But 1 or 2 persons who had taken the nosode, they did not get infected. So these kind of examples we have many, and all that together, they they established and rather validated my faith in the nosode that we made. Because you see, it was not a simple thing. There is always a doubt, there is always a question, because we do not have precedents. There are no many examples that we have seen where nosodes are used in pandemic or in major epidemic or endemics, right? We have heard about it. As I said earlier, we presumed that nosodes are useful, but we did not have much personal experience. At least I did not have much experience myself to see the efficacy of nosode as preventive measure in the larger community. And what you read in the literature about the previous pandemic 100 years back has its own limitations, because that data uh, was very interesting, but it has its own questions behind the data. So there are always questions, and of course, even our studies, they require a lot more studies, further research, to be able to, and we have to create much more data in order to substantiate the efficacy of the nosode in prevention. So having said that, one thing is was established with the limited preclinical and clinical studies that we made, that based on that, uh, we concluded internally that COVID nosode was safe and it was effective. And now was the time that we bring it to the masses. So as I said, uh, with my constant interaction with the government agencies, uh, I must now share with you a little good news also, that the government of India has shown a positive approach towards the use of the nosode, COVID nosode. And in the series of reviews that happened with the ministry and with the central government, where I was invited to talk and discuss and answer their technical queries, we are now at a stage that the nosode is undergoing the final stage review process at a probably at a very high level. And if we are lucky, and if the entire profession supports, then possibly there are chances that we should get some kind of government approval for the use of this nosode on the larger scale and on the larger population. So this is what I would like to share with you today. And yes, so it was a story of over 15 months in the making of the nosode, and that I would like to share with you very briefly. There are 4 research papers which are being written. 3 of them are either published or accepted in the peer-reviewed international journals, and the fourth one is still in the review process. Hopefully, they all will be published very soon, and they will be available in the public domain on Google, and we'll be able to share with you the links to these articles. So yeah, and of course we also conducted in between a homeopathic pathogenetic trial, that is a drug proving, on healthy volunteers, and that paper is also published. So you will very soon get an access to the symptomatology of the nosode as well. So this is in brief about the COVID-19 nosode, and which is now almost on its way to reach to the masses, and which I hope if we get through, then it should come in the hands of homeopaths in this country as well as in the hands of homeopaths across the world. So with this, I think I will conclude my talk, and I will invite questions from the participants and from the delegates listening to me. Thank you so much.
Thank you, Dr. Rajesh Shah. Now it is time for the question and answer sessions. I request our participant to go to the participant list and raise your hands so that we will be calling you based on the order of raising hands. You can directly ask questions or make your comments. The first is Dr. Sridhara Kurup. Sridhara Kurup, sir, you can unmute yourself and ask your question.
Dr. Rajesh Shah, are you able to hear?
Yes.
Audible?
Yes, you are audible. Please go ahead.
My sincere, hearty thanks to you. Done a very wonderful work. Thank you very much. First, my first question: whether we can use like that all other nosodes, we can use COVID also as a nosode?
Yes. In my view and in my experience with nosodes, COVID nosode will be just like any other nosode. And as you know, COVID virus is capable of inducing a very serious pathology at the level of, first of all, the immune system, at the level of the respiratory system, at the level of the cardiovascular system. It also produces symptoms at the level of muscles, nerves, brain, blood circulation. So that means that COVID nosode has a potential to be what is called as a polycrest remedy, because any nosode, or any virus or bacteria which has a capacity to affect a number of systems in the human economy can become a polycrest or a large medicine, like tuberculinum, for example, or medorinum, for example, and so on. So in my view, COVID nosode should be used not only as a preventive against the infection, but it can be used as a in those cases which are having the after effects of COVID. You can use this nosode. At the same time, you can use this nosode in the conditions not related to COVID at all. Like you are using tuberculinum for the patients of say, autism or migraine without having tuberculosis, isn't it? So as you can even use a nosode for a range of conditions. Of course, for that, we need to conduct more scientific studies in the coming time, which once we make it available to the profession, other researchers can also take up the studies, to prove the efficacy of the nosode for different conditions. I would like to say here that with the invention of the nosode, COVID nosode, we are entering into a new space in homeopathy. What I mean by that is that we will have to learn to look at the homeopathic medicines more scientifically, and we should conduct various systematic scientific studies to claim any efficacy of these medicines. So we must conduct several more studies in order to say that COVID nosode is effective. For example, in the cases of, say, pulmonary fibrosis, or COVID nosode is useful probably in the cases of rheumatoid arthritis, and likewise, based on the pathogenesis that it is capable of producing. I hope you are with me, you understand this concept. So that kind of law of similarity or similars that we have to apply to see what other possible role COVID nosode could have. So once it is made available in the hands of the homeopathic practitioners, I am sure the research-oriented homeopaths will be able to examine the efficacy of this nosode empirically in various conditions, and then we can have a pool of such sharing and experiences, whereby we can learn more about this nosode.